Usually when we talk about getting a medical diagnosis, there's this expectation of absolute precision, right?
Oh, yeah, like it's a math problem.
Exactly. It's almost like structural engineering. You break your arm, you go to the hospital, they take an x-ray, and you see that jagged white line on the bone. And the doctor just points at the screen and says, there it is, that's the problem.
Right, it's very comforting, very binary way of looking at health. It's either broken or not broken. You can literally see the structural failure with your own eyes.
But then you step into the world of neurodevelopment, and specifically aging. And suddenly, that x-ray machine is just utterly useless.
Completely useless, yeah.
We are looking at a diagnostic landscape that is incredibly murky. I mean, when we think about cognitive decline, losing our memory, our mental sharpness as we get older, we almost universally blame the brain itself.
We do. We picture the neurons just sort of getting tired, wearing out, slowly degrading after decades of use, which, to be fair, has been the bedrock assumption in neuroscience for a very long time. The brain was basically viewed as this isolated fortress, just sitting up there in the skull, protected by the blood-brain barrier, and just, slowly crumbling from the inside out as the clock ticks down.
But what if that fortress is actually being sabotaged from the outside?
That is the big question.
What if memory loss and cognitive decline aren't just about the brain getting older, but about the microscopic ecosystem living in your gut radically changing as you age?
That's a wild thought.
It really is. So, welcome to today's deep dive. We are looking at a fascinating Nature News and Views article by Yi-Ting Chang and Sarkis K. Mazmanian, and they're unpacking a truly groundbreaking study by a researcher named Cox and their colleagues.
And it is a dense study, but so rewarding.
Yeah. And the mission for our deep dive today is ambitious, but I promise is going to blow your mind. We are going to trace the exact step-by-step biological algorithm from a single gut bacterium traveling up the body's superhighways all the way into the brain's memory center.
We're basically going to map the whole route.
Exactly. We are going to see exactly how gut microbes can physically hijack your cognition. So, okay, let's unpack this.
To do it. It really is a paradigm-shifting concept.
Yeah.
The researchers essentially asked a very bold question. Can the microbes in your digestive tract actually dictate the age of your brain?
Which is a crazy premise.
Right. And to figure this out, they obviously couldn't start with humans. I mean, human lives are way too messy. They're uncontrolled.
Yeah.
So they started with mice. Specifically, they designed this behavioral case study that looked at what happens if you treat aging almost like a contagion.
Like a virus you can catch.
Exactly.
And this is where the setup gets a little wild. So they took young, healthy laboratory mice, and they put them in a cage to cohabit with aged mice.
Right.
Now, if you know anything about mice, you know they share absolutely everything in their environment. And by everything, we mean they ingest each other's droppings.
Yeah, it's a behavior called coprophagia.
Right. It sounds totally jarring to us, but for mice, it's a completely natural evolutionary way that they share and stabilize their microbiome.
And we should probably define that term clearly right up front because it anchors everything we're talking about today.
Good point.
So the gut microbiome is the entire sprawling community of microorganisms. We're talking bacteria, viruses, fungi, all living right there in the digestive tract.
Trillions of them.
Trillions. And because these young mice were living with the old mice and engaging in that shared environment, they rapidly developed an old microbiome.
Wow.
Yeah, their internal gut population shifted completely. It suddenly mirrored the exact bacterial composition you would find in an elderly mouse.
But physically, these young mice were completely fine, right? Like, they were healthy, their coats were shiny, they had all the physical markers of youthful energy.
Oh, absolutely. Physically, they were in their prime.
But when the researchers tested their cognition, their short-term memory had just completely tanked.
Which requires us to look at how scientists actually measure a tanking memory. Because, you can't just casually observe a mouse and say, yeah, he looks a little forgetful today.
Right. They aren't losing their keys.
Exactly. You need rigorous, validated behavioral tests. So they use two specific ones here to test different types of memory. The first one is called the Novel Object Recognition Test.
I love this one because it plays entirely on the fact that mice are just naturally curious, exploratory creatures.
It does. So imagine you put a mouse in a small enclosed arena with two identical objects, let's say 2 little wooden blocks. The mouse will run around, sniff them, explore them, and form a distinct memory of those blocks.
Okay, makes sense.
Then you take the mouse out for a bit, You swap one of those blocks for something totally new, maybe, like a little plastic cone, and you put the mouse back in. Now, a mouse with a healthy, functioning memory will spend way more time investigating the new plastic cone simply because they remember the old wooden block. They're bored of it.
So it tests recognition memory? But the young mice with the old microbiomes, the ones that shared the cage, they spent equal time sniffing both objects.
Right. They just couldn't distinguish the unfamiliar cone from the familiar block. Their recognition memory was just, well, gone.
And then there was the second test, right? The Barnes maze test.
Yes, which is much more intense because this one tests long-term spatial memory. Picture a brightly lit, elevated circular table with dozens of holes around the edge.
Sounds terrifying if you're a mouse.
It is. Mice instinctively hate open, bright spaces. They feel super vulnerable to predators. They want to hide immediately.
Naturally.
So only one of those holes on the table leads to a dark, safe drop box underneath. The mouse has to use visual cues posted around the room, like maybe a big black square on the wall, to remember where the safe hole is.
It's like building an internal map.
Exactly. Young, healthy mice usually learn this map really quickly. When you place them on the table, they just dart right to safety.
But the young mice with the old gut bacteria were just wandering around aimlessly. I mean, they were checking the wrong holes, exposing themselves to the bright light, completely forgetting the spatial map they should have easily formed.
Is a profound deficit.
But wait, I have to push back here as a skeptic. Could this just be the stress of having an older roommate?
Ah, the stress variable.
Yeah. I mean, maybe the old mice were hogging the food or, altering the social hierarchy. And we know severe stress ruins memory. How do we know it's definitely the gut microbes causing this and not just environmental anxiety?
And that is the exact critical thinking the researchers applied. They knew they had to rule that out. So to completely eliminate the stress variable, they removed the cohabitation entirely.
Okay, so no roommates.
None. They performed fecal microbiota transplants, often called FMTs. They took fecal matter directly from the aged mice, purified the microbes, and transplanted them directly into the guts of young mice.
Who are living alone.
Exactly. Living completely alone in solitary, stress-free environments. No old roommates, no social hierarchy changes, just pure microbial transfer.
And what happened to those solitary mice?
The exact same memory deficit appeared.
Wow.
Yep. The young, stress-free mice failed the novel object recognition test and the Barnes maze just as spectacularly. That isolated the microbiome as the definitive, undeniable cause of the cognitive decline.
Okay, so time for a quick 5-minute recap of what we've covered so far. Bullet 1. Sharing an old microbiome is enough to transmit measurable, severe memory deficits to young, otherwise totally healthy animals.
That's the core of it.
So to you listening right now, think about that for a second. If you could somehow swap your microbiome with a younger version of yourself, would you try it?
It's a very compelling idea. But, before we can even entertain that, we have to find the specific culprit.
Right, because the gut has trillions of bacteria across thousands of species. Which specific bug is actually pulling the strings to erase these memories?
The researchers couldn't just throw up their hands and say, well, old bacteria did it.
Yeah.
They had to get specific. So they ran a comprehensive genomic analysis.
Like a DNA map.
Exactly. They mapped the DNA of everything in the mouse gut over its entire lifespan, just watching how the population shifted month by month. And they found one specific villain whose numbers absolutely skyrocketed as the mice got older.
And what was the bug?
A bacterial species called Parabacteroides goldsteinii.
Parabacteroides goldsteinii, okay. But, correlation isn't causation, right? Just because it's there doesn't mean it's causing the problem.
You're exactly right. To prove it was the actual culprit, they did another experiment. They took a new batch of young mice and colonized their guts with only this specific bacterium. Nothing else from the old mice. Just P. goldsteinii.
And let me guess, it perfectly recreated the memory loss.
Perfectly. Just that one single bug was enough to break their spatial and recognition memory.
That is wild. But this creates a massive anatomical puzzle. How does a bug sitting down in the stomach or the intestines actually affect the brain? To figure that out, I guess we have to look at the brain's memory center.
Yes, the hippocampus. It's this small, seahorse-shaped structure deep in the brain, and it's absolutely critical for learning and memory formation. But we have to understand that the hippocampus isn't just one uniform blob of tissue.
Right.
It has highly distinct processing subregions. You can think of it kind of like different specialized departments in a manufacturing factory.
I love that analogy, a memory factory.
Yeah, and the researchers look specifically at 3 regions, which are called the dentate gyrus, the CA3, and the CA1.
Okay, let's visualize those departments for the listeners. So the dentate gyrus is kind of like the receiving dock of the factory, right? It takes in all the raw, new sensory information from the outside world. Then you have CA3, which is like the pattern matcher.
Yeah.
It's associating that new information with older memories. And finally, CA1 is the output department, packaging the final memory and sending it back out to the cortex for long-term storage.
That is a perfect way to look at it. And to see if these departments were actually working in the mice, the researchers didn't just look at the physical tissue. They looked at the genetic activity happening inside the neurons themselves.
Like down to the molecule.
Yes. Specifically, they looked for the presence of a protein called CFOs. Now, CFOs is what neuroscientists call an immediate early gene.
Right. I always picture CFOs as the literal light bulb switching on in the brain. Like the exact millisecond a memory is made or a neuron fires to process a new environment, CFOs is produced instantly. The light goes on.
Exactly. It's the visual marker of activity. So when the researchers looked at the brains of the young mice colonized with the aged microbiome, what do you think they saw?
Darkness.
Total darkness. The expression of CFOs was drastically reduced across the board in the CA1, the CA3, and the dentate gyrus. The neuronal activation had essentially plummeted. The entire memory processing factory had just powered down.
So in the mice with the aged microbiome, those light bulbs in the CA1 and CA3 rooms are suddenly very, very dim.
Yeah.
But here's the part of the study that gave me genuine hope when I read it. It is completely reversible.
It really is. The damage isn't permanent structural decay like we used to think.
So what did they do?
Well, they took aged mice that naturally had poor memory and naturally high levels of P. goldsteinii, and they gave them broad-spectrum antibiotics to just wipe out the gut bacteria entirely.
That's a total reset.
Right. And alternatively, they raised other aged mice in completely germ-free, sterile bubbles from birth, so they never had any microbes at all.
And the result?
In both of those scenarios, without those specific age-associated microbes present, the memory deficits in the aged mice were corrected. Their cognitive function actually improved back to youthful levels.
That is unbelievable. Okay, time for another quick recap bullet. A specific age-associated bacterium, P. goldsteinii, directly reduces the cellular activation, those CFOs light bulbs, in the hippocampus memory centers. And if you wipe out the bug, the memory factory powers back up.
That's the mechanism at a high level, yes.
So, to the listener,
Yeah.
next time you walk into a room and completely forget why you went in there, you really have to wonder, is it just a random brain glitch? Or are your gut bacteria actively throwing a wrench into your hippocampus?
Which brings us to the core mystery of this entire deep dive, the physical mechanism itself.
The how.
Right. Because the gut and the brain are physically very far apart. They're separated by dense tissues, organs, and of course, the highly restrictive blood-brain barrier.
Which is designed specifically to keep pathogens out of the brain.
Exactly. So how is a microscopic organism trapped down in the intestines turning off the lights inside the skull?
This is where we need to trace the exact biological algorithm.
Yeah.
Seriously, if you're listening, grab a mental notepad, because we are going to build this chain reaction right now, step by step. And it all starts with the bacteria's exhaust fumes.
That's a great way to put it. Step 1. As P. goldsteinii goes about its life in the gut, fermenting and digesting the food you eat, it produces metabolic byproducts.
Right.
Specifically, it churns out molecules called MCFAs. That stands for medium-chain fatty acids.
MCFAs.
Yes. You can think of these MCFAs as chemical smoke signals that the bacteria are constantly releasing into their surrounding environment.
Okay, so the bacteria are pumping out these MCFA smoke signals in the gut, but they don't stay there, do they?
No, they don't. Step 2. They slip through the intestinal wall and enter the bloodstream. Now, circulating in your bloodstream are your immune cells, specifically a type called myeloid cells.
Let's call the myeloid cells the body's security guards. They're constantly patrolling the blood for signs of trouble.
I like that. So on the surface of these myeloid security guards are receptors. Receptors are essentially complex molecular locks that only open when a very specific chemical key fits into them.
Okay.
The receptor we care about here, the specific lock, is called GPR84.
GPR84.
Right. And it just so happens that the MCFAs floating in the blood, those smoke signals from the gut, are the exact molecular key for the GPR84 lock. So the MCFAs bind to the myeloid cells.
Okay, moving to step 3. The smoke signal has been officially received by the security guard. The key is in the lock. Once that GPR84 lock is turned, it sets off a massive alarm system inside the myeloid cell.
Yes, and this alarm system is a multi-protein complex called the NLRP3 inflammasome.
NLRP3 inflammasome. Listeners, you don't need to memorize that alphabet soup. Just think of it as an intracellular panic button.
A panic button's perfect. When that panic button gets pressed, it drives the myeloid cell to start pumping out heavily inflammatory signaling molecules called cytokines.
Fire alarms.
Exactly. The two major ones produced here are IL-1 beta and TNF. These cytokines are the body's chemical equivalent of a blaring, deafening fire alarm, just flooding the bloodstream with inflammation.
Okay, so now we have these inflammatory alarm bells flooding the area, but they still have to reach the brain. How do they do that?
That's step 4. They do this by physically interacting with the nervous system, specifically the vagus nerve.
Ah, the vagus nerve. The great transatlantic cable of the body.
That's exactly what it is. It's a massive, complex bundle of nerve fibers running all the way from the digestive organs straight up into the brainstem. The researchers found that the inflammatory cytokines specifically target sensory neurons on the vagus nerve.
No, wait. Normally, you might think an inflammatory alarm would stimulate a nerve, right? Like making it hyperactive or sending a pain signal.
You would think so. But in this specific case, it does the exact opposite. The surge of inflammation acts almost like a tripped circuit breaker. Yeah, it is so intense that it forces the vagus nerve to shut itself down just to protect the system. It induces a profound nerve block, effectively cutting the communication line from the gut to the brain.
Wow. Which brings us to step 5, the final domino. Because that communication line is cut, the normal, healthy background chatter that usually travels up the vagus nerve to the brain suddenly goes dead quiet.
Eerily quiet.
And this unnerving silence reaches a vital relay station in the brainstem called the NTS.
Right, the NTS is the gateway. If the NTS receives a suppressed, quieted signal from the vagus nerve, its own activation drops. And because the NTS feeds directly into the memory centers, that suppression cascades right into the hippocampus.
Resulting in those dimmed CFOs light bulbs we talked about earlier.
Exactly. The factory shuts down because the power line got cut.
Okay, let me make sure I have this whole biological domino effect straight. Bullet recap time. The bacteria makes fatty acids. Those fatty acids bind to the security guard immune cells. This triggers the panic button, which releases massive inflammation. That inflammation trips the circuit breaker on the vagus nerve, silencing it. And that silenced nerve quiets the brainstem, which ultimately turns off the lights in the hippocampus.
You nailed it. It is a masterclass in cross-system biology. We just watched a cascade originate in the microbiological system, travel through the immunological system, and ultimately disable the neurological system.
It's terrifying, but also kind of beautiful.
It really is.
So looking at this entire chain reaction, I want to ask you, the listener, if you were a scientist, where would you try to break the chain to save the memory? Which step feels the most vulnerable to you?
And, the researchers asked that exact same question. They wanted to test the algorithm by systematically breaking every single link in that chain to see if it would rescue the mice's memory.
And the results are what make this study so legendary. Wait, tell them what happened when they altered the immune cells.
Okay, so first, they genetically engineered a group of mice to completely lack that GPR84 receptor on their immune cells.
So they removed the lock.
Exactly. The bacteria were still producing the fatty acids, but the lock was gone. The key had nowhere to dock.
And the result?
The mice kept their memory perfectly intact, even with an aging microbiome.
That is incredible. What about the panic button, the inflammasome?
Same result. They used mice that were genetically deficient in that inflammasome panic button. Without it, no inflammatory cytokines were produced, the vagus nerve never shut down, and the memory was completely protected.
Wow.
And perhaps the most elegant intervention of all, they used targeted technology to artificially force the vagus nerve to stay active, completely overriding the inflammatory block.
They just taped the circuit breaker open?
Basically.
Yeah.
And once again, the memory was completely saved. Every single intervention proved the algorithm was correct.
Okay, so I have to ask the obvious million-dollar question here.
I think I know what it is.
If we know the exact bug and we know the exact pathway, do we just create a probiotic pill to eradicate P. goldsteinii? Or like a drug to block that immune receptor, and suddenly we cure severe memory loss disorders in humans, like Alzheimer's or frontotemporal dementia?
This is where we need a very necessary, very grounding reality check. We always have to remember the context of the data.
Right, it was in mice.
Exactly. Lab mice live in highly controlled, pathogen-free environments. They eat the exact same sterilized chow every single day of their lives. Their microbiome aging in this study is purely a consequence of the host aging.
Meaning what, exactly?
Meaning, as the mouse's immune system naturally weakens or its metabolism changes with age, it selectively allows that specific bug, P. goldsteinii, to overgrow.
Right, whereas humans are basically walking ecosystems of chaos.
We really are.
I mean, we eat wildly different things from day to day. We travel across the world. We take different medications. We catch random viruses. Our microbiomes are constantly under siege.
Exactly. The leap from a lab mouse to a human is vast. In fact, the source article points out a fascinating contradiction in the current scientific literature.
Oh, what is it?
Well, while this specific mouse study clearly shows P. goldsteinii causing memory loss, there are human studies that show the relative abundance of P. goldsteinii actually decreases with age in some people.
Wait, it goes down in humans. How does that make any sense if it's the bug causing the aging?
It proves how unimaginably complex microbiology is. See, bacteria aren't just one uniform monolithic species. Think of it like dogs. All dogs are the same species, but a Chihuahua and a Great Dane behave completely differently.
That's a great point.
Bacteria have different strains. The specific strain of P. goldsteinii that overgrows in the lab mice might be highly inflammatory and produce those specific fatty acids. But a completely different strain found in humans might be benign or even actively anti-inflammatory and protective.
Oh, wow.
Yeah. This is why we urgently need human proof-of-concept studies to look at specific bacterial strains, not just the broad species level.
But this doesn't mean the study is a dead end. Not at all.
Well, absolutely not.
Even if the exact bug is different in humans, the pathway, the proven idea that gut metabolites can trigger an immune response that suppresses the brain, is a massive breakthrough.
It changes everything about how we view neurodegeneration.
Right. And it brings us back to a very practical, everyday application for anyone listening right now: diet.
Precisely. We know that old age and genetic risk factors are the primary drivers of severe dementias. But diet and lifestyle are incredibly powerful modifiers of cognitive decline.
The source text explicitly calls out modified Mediterranean-style diets. These are diets high in fiber, rich in polyphenols from plants, and full of healthy fats like olive oil. Early clinical studies show they hold real, measurable promise for protecting our memory as we age.
And here is the brilliant twist that this research provides. For decades, we assumed the Mediterranean diet worked because those nutrients were directly feeding the brain tissue, or maybe just protecting the blood vessels in the skull. But based on this gut-brain algorithm, the protective effects of a healthy diet might actually be working because they change the microbiome.
That is profound. You aren't just feeding yourself, you are selectively feeding a specific ecosystem of bacteria.
Exactly. A high-fiber diet favors bacteria that produce neuroprotective chemicals, essentially starving out the bacteria that produce those inflammatory fatty acids we talked about.
You are literally gardening your gut to protect your hippocampus, which is an empowering thought. But, let's be realistic. Sticking to a strict, perfect diet for decades is incredibly hard for the vast majority of people. I mean, life gets in the way.
It does. Adherence to long-term dietary interventions is one of the biggest barriers in modern preventative medicine. It's just tough.
Yeah.
But that is exactly why mapping this microscopic gut-to-brain mechanism is so vital. If we can't get everyone to eat perfectly all the time, but we understand the exact locks, keys, and alarm systems involved in cognitive decline, we can develop actionable, long-lasting medical treatments.
Like a bypass.
Yes. Imagine a medication that gently blocks that GPR84 receptor on your immune cells. It would essentially mimic the biological effects of a perfect diet, shielding your brain from microbial inflammation, regardless of what's happening in your gut.
So to pull all of this together for our final recap, while human microbiomes are vastly more complex and messy than those of lab mice, mapping this specific gut-immune-brain pathway opens a massive door. It paves the way for targeted treatments that could mimic the cognitive benefits of a perfect diet, protecting us against the very mechanisms of dementia.
The leap from a simple behavioral observation mice sharing a cage to a granular receptor-level biological algorithm is a triumph of modern science.
Yeah.
It fundamentally forces us to reconsider where the boundaries of the brain actually end.
It really does. Which leaves us with a lingering thought to close out today's deep dive. Throughout human history, in philosophy and science, we've always viewed our memory, our mind, our conscious recall as the absolute core of who we are.
The foundation of identity.
Exactly. It is the foundation of our identity. But if our cognitive sharpness, the very light bulbs of our memory, are heavily dictated by the shifting populations of microscopic bacteria living in our intestines,
Yeah.
how much of our mental aging is truly us, and how much are we simply at the mercy of our microscopic passengers?
That is a profound question that really challenges our sense of self. The old X-ray machine for cognitive decline might be completely broken, but we are finally starting to build a new lens to see the problem clearly.
And that's exactly what we try to do here. Thank you for grabbing that mental notepad and joining us on this incredibly complex journey from the gut to the immune system, up the vagus nerve and into the brain. Keep feeding your good microbes, keep asking the big questions, and we'll catch you on the next deep dive.